Hope you are keeping fine at your homes. Welcome to another video lecture on generation of humoral immune response. We have already seen that when an antigen, a pathogen, a bacteria, a virus, a fungi, any foreign particle, even a foreign protein, when enters the body reacts to it in a specific manner. This specific reactivity induced in a host by an antigenic stimulus is known as immune response. This response can be initiated by the immune system in two ways. They are cellular immune response and humoral immune response. In today's lecture, we shall focus on the humoral branch of immune response, especially the mechanism and the effector functions. So we shall see what is humoral immune response. Host defenses that are mediated by the secreted antibody which are present in the plasma, lymph and tissue fluids. So it is clear from the definition that the antibodies that are secreted by the B cell and which are present in the fluid part of the body, or the antibodies that are present outside the cell within the body, is responsible for the humoral branch of the immune response. And therefore this is also called as antibody mediated immunity. The Latin word humor, which means fluid, from which the word humoral branch or the humoral immune response is coined. The humoral immune system deals with antigens from pathogens that are freely circulating or outside the infected cells. So now we shall see what are the important cells involved in initiating humoral immune response. The first we have here the B cells. So B cells only are the main cells that is involved in case when the antigen that is entered is T cell independent. So T cell independent antigens, this is an immunogen that stimulates B cells to synthesize antibodies without the help of T cells. Why they do not require the help of T cells? Because these antigens are less complex. For example, the carbohydrate antigens or the nucleic acid antigens, these are examples for T cell independent antigens. Now we have the second cell types, that is antigen processing and presenting cells and also T helper cells. And this antigen processing and presenting cells here can be a B cell. If it is some other cells, then B cells also are involved here. That is in case of T cell dependent antigen. So these antigens are mainly protein antigens which are more complex in nature. So now we shall see the mechanism of production of antibody. This diagram here, the T independent antigen, how it interacts with the B cell, generating the two basic signals that is required for the B cell to activate, proliferate and differentiate into antibody producing plasma cells. That is here the antigen binding to the receptors. You can see here the antibody, they are the membrane bound antibodies present on the B cell that act as antigen binding receptor. Once this antigen binds to this receptor, that itself will initiate the first and the second signal that is required for the B cell for activation. And here you have the other type, T-dependent antigens. That means already we have seen that T-dependent antigen, that means it requires the help of T helper cell to initiate the humoral branch of the immune response, that is to produce antibodies. So here you see this is the B cell, and it can be any other antigen presenting cells also, B cell or any other antigen presenting cells. And since it is B cell, it has got its receptor, that is the antibody. This is called the membrane antibody, and they are interacting with the antigen. This is the antigen binding site on the antibody molecule, which is acting as the receptor. Once this antibody or the receptor binds to the antigen, that this antigen enters by endocytosis. Then since B cell is also one of the professional antigen processing and presenting cells, now this antigen that is entered will be processed and presented to the T cell. See, this is the class 2 MHC molecule, since this is the T helper cell, and the professional APCs, they express class 2 MHC molecules. The processed antigen is presented along with the class 2 MHC molecule, and this interaction will provide the signal required for the activation of T helper cells. A little more detail on how the T-dependent antigens initiate the humoral response and finally leading to the production of antibody. Here you can see the antigen. Here, for example, it is a bacteria. This is the B cell receptor and this is the B cell. So here the B cell is also an antigen presenting cells. So this binding will facilitate and the bacteria will be inside the cell, and this will be processed and presented to the T helper cells. Here it is the TCR, the CD4. This CD4 helps the interaction of this TCR with the MHC antigen complex. So once this interaction will take place, in the previous slide we saw that the secondary signals also the T helper cells will receive for the activation, and due to the activation T helper cells produces these cytokines, and these cytokines now will activate the B cell, and this activates and it enters into the division cell cycle, divide into number of cell cycles, forming a clone of cells. So it's called clonal expansion due to cell proliferation. So now these cells that are produced now will differentiate into two different types of cells. What are they? Here you can see, here it is the plasma cell that you cannot see, and here it is the memory cells. The plasma cell wants activation, proliferation and differentiation. So here is the effector cell is the plasma cell. This is the plasma cell, and this plasma cell has the ability to synthesize the antibodies that will be acting as an effector molecule. And that will be in the first case when the antigen enters the primary response. Antibody is produced. Now this antibody has to perform the effector functions. Now here you can see some of the cells that are produced will differentiate into memory cells. These memory cells will play a role in the secondary immune response, which means when the same type of antigen enters the body, after some time, these cells will recognize these antigens and mount an immune response in a very quick manner. So here, this diagram shows another important characteristic features of the humoral immune response, that is class switching. Class switching is also called isotype switching. So what is this isotype switching? We know that already that there are different classes of antibodies produced by B cell on activation. So there are 5 classes: IgM, IgG, IgA, IgD and IgE. So class switch means there is a switch between the production of different types of antibody. Generally, when the activation, this process already we have seen, the activated T helper cells and secret cytokine. So these cytokines you can see interleukin 4 is there, interleukin 5 is there. These are cytokines. You see, once the T helper cells gets activated and secretes the interleukins, different types of cytokines are secreted, and that will actually determine how the process will proceed further. The normal course of the activation and differentiation process, what is happening here? You can see this is the B cell with the T helper cells products activating, it is interacting with each other and activating, and also the cytokines are there. These differentiate into plasma cell. You see the plasma cell now producing IgM antibody. So we know that IgM antibody is a pentamer. So this is the antibody type that is produced in the initial stages. So now there is a class switch, that means a change from the type of antibody that the B cell produces. So this is called the class switch or the isotype switching. You see, in the presence of interleukin 4, suppose this cytokine activated T helper cells producing interleukin 4. So in the presence of interleukin 4, you see what type of switch is taking place? From IgM, the IgG antibody and the IgE is produced. And at the same time, if cytokine interleukin 5 is present, then there is a switch from IgM to IgA production. So there is change in the production of antibodies, and this is called class switch. And the different functions, effector functions are there for different types of antibodies that is produced. So now we shall see the steps involved in the T cell independent antigens. Once the T cell independent antigens enter the body, see, these are the antigens. See, there are different variety of B cells. Now here 6 different types of cells are shown. That means these cells are specific for different types of antigens. The antigen that has entered the body specifically recognizes this B cell. That means the B cell receptor on the surface of this B cell is able to recognize these antigens. That means it is able to attach with this antigen binding site of this receptor, can bind to these antigens. But these antigens are not able to bind to this or this or this, any of these except this. So that shows the specificity of the immune response. So once this interaction has taken place, if you minutely observe, you can see the differences in the variable region or the antigen binding site. These points, if you follow, you can see the differences are minute. The differences will be not so much, it will be very minute differences, and that differences is sufficient to differentiate different types of antigens and to respond to differently. See, once this interaction takes place, now use the cell. Because it is T cell independent, therefore this interaction will now lead to the activation of B cell directly. So activation, that means now next it will lead to the proliferation. B cell activation, antigen binding to naive B cell receptor. Clonal selection, division of that activated B cell. That is what we can see here. The first generation, then second generation, third generation, it follows a number of generations. Once the sufficient clone of cells are produced, then the cell will enter into the differentiation phase. This is the step 3 here, differentiation into plasma cells and memory cells. You can see here, these are the cells, number of cycles divided, and some of the cells, they are producing memory cell and some of the cells plasma cells. These are the cells now important for the primary immune response, because now this plasma cells will produce antibodies. These antibodies are secreted into the blood. So this can reach to any part of the body wherever the antigens, similar type, same type of antigens are present. It can bind to that antigen and then it can destroy that antigen with the help of complement. So due to the isotype switching, and different types of antibodies are produced which has got different effector functions. So IgM antibody is good at fixing complement and very good at grabbing pathogens. And IgG, they fix complement and also mediate ADCC, antibody dependent cell mediated cytotoxicity. IgA, they neutralize toxins and pathogens, and they are present in secretions like saliva, tears and so on. IgE, they are involved in inflammatory reactions and also in some of the parasitic infections. In such cases, IgE antibodies produced by the plasma cells have different effector functions. The most important ones are virus and toxin neutralization. So what is this virus and toxin neutralization means? See, virus has got the infective sites which binds to the host cells, and the toxin also has the binding site, and when the produced antibody is produced against these viruses or toxins, these antibodies bind to this virus or the toxin, neutralize them, which means that it binds to the infective site and block the binding site. Therefore, this virus or the toxin which is not able to bind to the host cells. So unless it binds to the host cell, it cannot cause disease, thereby preventing the virus from causing disease and protecting the body from the disease. And secondly, the another function is the opsonization. So that means antibody is produced. That also IgG antibody is the protein opsonin which coats the surface of the pathogen, making it more susceptible to phagocytosis. So these are phagocytes, for example here macrophage. This has got Fc receptors on the membrane, and the antibody coated antigen comes in contact with cell. It facilitates easy binding to this cell. Because of this, these phagocytes is able to, through the endocytosis, can take in these antibody pathogen complex. Once they are inside the body, they will be broken down into fragments and eliminated. So this is one of the another important function of antibodies. And the third function you can see here, complement fixation and formation of the membrane attack complex. So you can see here the tumor cell. So it can be any infected cell also. So against the antigens on these cells, these antibodies are produced in the body as a result of immune response. So this antigen, once they are produced, they bind to this target cell or the antigens. So once these these are antigen and this antibody, they form an antigen-antibody complex. So once this complex is formed, this complex is able to activate complement components. So complement components are present in the serum. Whether antigen enters or not, it is there, produced by the liver cells. But if it has to function or become active, it needs the help of antigen-antibody complex. So this complex now is therefore able to activate. So they are numbered, complement components are numbered C1, C2, C3, C4 and so on. So the activation process starts with the C1 and in a particular sequence, the different components of the complement gets activated and finally that will lead to the formation of MAC, that is membrane attack complex. Once this complex is formed, this complex get attached to the membrane and get inserted into the membrane, forming pores of different sizes. So once the pores are produced on the membrane of the target cell, the tumor cell or the infected cells, then the lysis of the cell will take place. Therefore, in this way, the antibody is helping the destruction of the infected cell, either virus infected cell or the transformed cell, which is a tumor cell. And lastly, you can see here, antibody involves in ADCC, that is antibody dependent cell mediated cytotoxicity. So here antibody is acting as a mediator only. You can see here, this is the cell which is responsible to kill this cell. But this cell cannot kill this cell without the help of the antibody. And what does the antibody do? Antibody binds to the target cell and helps this infected cell or the tumor cell to come in contact with cell. So that means these are the infected cell, antibody is produced against it. With its antigen binding site, it has bound to this cell, and now the Fc portion of this antibody is free. So with this site, there are Fc receptors on the NK cell, that is natural killer cell. So both antibody binds to this cell as well as the NK cell. So this brings the NK cell closer to the infected cell or the tumor cell which has to be destroyed. And this is the role the antibody plays. Once they are brought close to each other, then the natural killer cell will destroy the cell. Destroy this infected cell or the tumor cell. So that way it will protect the body from either developing the disease or from developing tumor cells. So these are some of the effective functions the antibody carries out in the body. And to summarize the humoral immune response, from the beginning, antigen interacts with the B cell receptor on B cell or receptor on APC. Antigen is processed and displayed on the surface of APC or B cell by a special receptor protein called MHC2 for recognition by helper T cells. B cell is activated by the helper T cell to divide and produce secreted antibodies. Antibodies circulate in the serum and lymph and bind to antigens in the serum and lymph, leading to its destruction. Each B lymphocyte makes a unique antibody molecule, and over 1 million different B lymphocytes are produced in each individual. So each individual can recognize more than 1 million different antigens. And some B cells become memory cells. They respond quickly when the same type of antigen is encountered again, leading to secondary immune response. Hope this video will be useful for you. Thank you. God bless you.